So in this video we're going to look at the way to perform a urinalysis to give some interpretation. So urinalysis is one of these tests that it is simple to make and it gives so much information before performing other tests. And this test specifically, of course, is directed to study problems in the urinary system. So we will be doing this urinalysis with a dipstick of this multi-sticks 10G of Siemens, and then it has different components. It has 10 different tests, and all of these different tests are for the following substances or parts. So we will study for the presence of leukocytes in urine, nitrites, urobilinogen, protein, pH, blood, specific gravity, ketone, bilirubin, and glucose. So if the test gives different results for the normal, which are here, then you can give a specific interpretation. So in terms of leukocytes, we shouldn't have leukocytes in our urine because urine is supposed to be sterile. So it shouldn't have any microorganisms. So if someone has leukocytes in their urine, that means that they have an infection. Nitrites, it's a component of these byproducts of ammonium. So we shouldn't have any of them. So it's supposed to be negative. Now, if someone has these nitrites, it can be a degradation product of the epithelial cells of the urinary system because someone has an infection, and these proteins that makes these urinary system cells are being degraded. Now, for urobilinogen, this is a normal component in the urine, so we're supposed to have between 0.2 and 1 milligrams per deciliter, and this is the pigment that actually gives the urine this yellowish color. And then for proteins, we shouldn't have any proteins because the filtration membrane that exists in the kidneys, which is the glomerulus, it's supposed to be completely sealed. So nothing should be of proteins in the urine, no proteins in the urine. And if there is proteins in the urine, that means that the person has glomerulonephritis. For pH, it's supposed to be acidic between 5 and 6 because as we are metabolizing our nutrients like glucose, we generate organic acids that ultimately get excreted through the urine. And when someone has basic pH like over 6.5 or 7, it's alkaline urine, so that means that the person will have an infection of the urinary tract, especially by Proteus, which is a bacteria that converts urea into ammonium, and ammonium it's a component that is basic. Now, blood, it shouldn't go into the urine as well, so we shouldn't have any traces of blood. And if someone has blood similarly as to when they have proteins in their urine, that means that they have glomerulonephritis. Or it can have the person, if it's actually hematuria, in which you have the urine completely red, the person might have a kidney stone. Now, for a specific gravity will be how dense is the urine, so it shouldn't be so diluted, but shouldn't be so concentrated. So the normal specific gravity is between 1 and 1.05 and 0.05. And if someone has over diluted urine, that means that it has a problem with ADH, which is called the antidiuretic hormone. For ketones, we shouldn't have any because this is a byproduct of the metabolism of these amino acids. And if someone has ketones in their urine, that means that the person is either starving or it is undergoing ketosis. Bilirubin, it's a pigment, so it is part of the metabolism of the heme. so we shouldn't have any bilirubin in our urine, and also we shouldn't have any glucose. Our body tends to save any of the nutrients, so if someone has glucose in their urine, more than likely will be someone who has diabetes, or it can be a pregnant woman, because at some point the threshold for glucose retention in the pregnant woman, it's a little bit different than someone who's not pregnant. Now, for reading these results or this test in the urinalysis, you will start reading from glucose, which you can start reading 30 minutes after you have dipped this test strip into the urine sample, going up into leukocytes, which can be read after 2 minutes after you have dipped this strip into the urine sample. So now let's go here into the countertop so we can see how the urinalysis is performed. So in here we have the actual strips for urinalysis, and then in here we have these samples of urine. So we have one that is called normal sample. This is abnormal A and abnormal B. Now these strips that you see here has 11 squares. This upper square is just the reference line of where we are going to start reading over this flask. So basically when you're reading, you align this top square to this one in here, and then you will move in this direction, aligning the rest of the squares until you reach the last one. So you start again reading from glucose, which is found in here, in this part, and then you continue to bilirubin, ketones, specific gravity, blood, pH, proteins, urobilinogen, nitrites, and leukocytes. So you're finishing there. Now, towards the bottom of the flask, you have the negative results or the normal results. And if you see a change in color after you dip the strip into the urine sample, what you will do is to scroll each of these into the corresponding key, so you can read what were the changes. Now, in order for us to know which strips will correspond to which sample, we use the Sharpie to mark here on one end what sample was. So with this one, I put the A because it's for abnormal A. For this one of normal, I'm going to put an N. And then for this one, for abnormal, I'm going to put a B, abnormal B, because, a B because it's abnormal B. Now, when you're done, you can either leave it this alignment, and then you can take a picture so you can keep a record of your results. And at some point, what you will do just to test for this urinalysis is just grab this strip by the edge, dip it inside the test tube, drag it immediately out, and you have to cover all your squares with urine, and then put it here on one side, and then you can start reading. So this is what then we're going to do right now for each of the samples. And I'm going to open them all right now. And then I will start doing the test. We're going to get a little bit more time for reading it because I'm doing 3 samples at once. At some point, even if you delay a little bit in the reading, it shouldn't change that much. Okay, so this is how they look like. And in order for us to start reading, what you can do then it's just get rid of the excess of urine, just put them here on one side. And these are artificial urine samples, okay? So I start here on abnormal A, and if you see, it has this brown color for glucose. So if I scroll up, I can find that this abnormal sample has 2,000 milligrams per deciliter of glucose. So basically, it's 2 grams of glucose per deciliter, which will be 100 milliliters. So that's a lot. Okay. So what can I interpret in here is that possibly this sample comes from someone who has diabetes. Now for bilirubin, it has this change to this kind of from yellow to rose color or a little bit darker color. So it's a small trace of bilirubin in there. And yeah, you can think about that someone has then a little bit of problem in the liver as well. And then for ketones, it has gone into a color that it doesn't even correspond to here. So what does that mean? So it can mean that the person has more ketone body than even you can detect in here. So this is possible because since it has a lot of glucose in it, so you expect that the person is the sample is coming from a person who has this ketoacidosis. So well, actually, never mind. This is for this. So here, so this is the third strip. So it will go up to here. So that's for ketones. So it has traces. So at some point, yes, you can expect some traces of ketones in someone who has diabetes. And now for the next one, a specific gravity. So a specific gravity, it's 1,000, so and 5, so it's normal. Traces of blood, you don't see it, so it's negative. That's good because that means that this person sample, it doesn't have blood, so possibly there's no damage still to the kidneys. pH is 5, so that's good, it's acidic. And then for proteins, it's negative, so that means as well that the kidneys are functioning well. There is no glomerulonephritis. Urobilinogen, it's normal, 0.2, and negative for nitrates and negative for leukocytes, which at some point, that's strange because usually when someone has this high amount of glucose in their urine, you expect to have leukocytes or an infection because bacteria, UTI bacteria like to use glucose for their replication. Now for the normal sample. Okay, so you have negative for glucose, negative for bilirubin, and then the next will be negative for ketones. You can see a little bit of this green color in there, but this might be part of these chemicals that has run from this square into this. Now for specific gravity, it's 1.05, negative for blood, negative for, pH 5, negative for proteins, and then urobilinogen, that's 0.2, negative for nitrates, and negative for leukocytes. So that looks good for the normal. Now for sample abnormal B, so it's negative for glucose, negative for bilirubin, negative for ketone. A specific gravity is 1.00. Blood, it has some tiny green dots in there, so it can be in between trace or moderate. And then the pH, it is so basic, so it's 8.5. And then for proteins, has trace. And then for urobilinogen, it's normal, 0.2. negative for nitrates, and kind of negative for leukocytes. So the only thing in here is a little bit of, let me align it again. So it's a little bit of blood, okay, which is this. So at some point in here, together with this pH, which is very basic, can pinpoint to someone having glomerulonephritis and having this urinary tract infection that you cannot see here with these leukocytes, but it can be a produce infection because you have this change of pH from acidic 5 until very basic. So remember, produce, it's this bacteria that converts urea into ammonium. So anyway, so this is all for the urinalysis video. I will see you in the next.